Welcome to the session on From Species to Ecosystems, Integrated Population and Speciation Genomics. I'm Richard Durbin and I'm co-chairing with Nina Therkelson. So we've got 5 talks from Axel Jensen, Rosalia Pinheiro, Blessing Nwachukwu, Jeff Lozier, and Catherine Farquharson.
And please go ahead and add your questions to the Q&A box, and remember to write the name of the speaker that your question is for, then we'll have a chance to address them at the end. And also for the next hour, we'll be focusing on the talks here, but we wanted to also just make sure that you know there's a set of really great posters associated with this symposium too, so we encourage you to check those out as well. So let's hear this great lineup of talks.
So thank you very much for having me. My name is Axel Jensen and I am a PhD student at Katarina Gushansky's lab at Uppsala University. And I will talk a little bit about my ongoing project about the prevalence of ancient hybridization in an African primate radiation. So hybridization was historically considered more or less an evolutionary dead end, since genomic incompatibilities in the parental species tend to cause some problems in the offspring, like in this left example here, where hybridization between 2 different swordtail fish species produces this tumor overgrowth phenotype in the hybrids. It's widely recognized today, however, that hybridization can also act as an evolutionary catalyst, as in this cichlid example, where introgressed alleles were found to play an important role in the adaptive radiation of this Malawi clade here in orange. So while this is an increasingly studied topic, due to the large genome sizes of mammals, those are still fairly understudied in this respect. So this is where our system comes in, the Guenons. So the Guenons is a very species-rich group of African primates comprising over 60 species and subspecies across 6 different genera. And these are further subdivided into 12 different species groups. And in addition to a wide morphological and ecological diversity, they also harbor this astonishing karyotypic diversity with diploid chromosome numbers ranging from 48 to 72. And what's even more fascinating is that in spite of all this diversity and, seemingly barriers, they still manage to hybridize and seemingly produce viable offspring and fertile offspring. So this is something that we wanted to look closer at and see if this might have played a role throughout the evolution of Guenons. So to do this, we generated high coverage whole genome sequencing data from 23 different Guenon taxa, representing all the genera and all the species groups. And we called nuclear SNPs against the rhesus macaque reference genome. And we also assembled the mitochondrial genomes for all the included samples. And the first thing that I want to show you is the nuclear topology. So what you're looking at now is the topology constructed only using autosomal data. And I mentioned before that they have this ecological diversity, and on the large scale, you can divide them into 2 major groups as to whether they are mainly living arboreal in the trees or being more terrestrial. And in the nuclear topology, these 2 groups come out as monophyletic, with 7 species group being predominantly arboreal and 5 being more terrestrial. When we also construct the phylogeny using the mitochondrial data, however, things start to become a little bit more complicated. So there are several cases of mitonuclear discordances, even scenarios where you have species shifting position from the middle of the terrestrial clade to the middle of the arboreal clade. So clearly, mitochondria have been shuffled around throughout this radiation. And the most plausible scenario is that this is involving introgression to some extent, and possibly in combination with incomplete lineage sorting. And the previous PhD student in our lab, Tom van der Walt, looked specifically at one of these events, this one that you see down here, so involving the Dryas and the Pidgeriters clades. So looking at this mitochondrial introgression, he was able also to find nuclear introgression corresponding perfectly to this introgression event. So taking off in these mitonuclear discordances, we wanted to look at the entire radiation and see if we would be able to detect nuclear introgression events, and if we could explain these mitochondrial capture events with nuclear introgression as well. And to look at introgression, we used the D-statistics. So the D-statistics is simply a test for incongruent allele sharing patterns in a 4-taxon topology looking like this. So you have an outgroup, which in our case was always set to the macaque. And then you have a P1 and P2 being sister species and a P3 being a midgroup. And a significantly positive D statistic is then indicating gene flow between P3 and P2. So what you see here is simply all those trios in our data sets that showed a significantly positive D statistics. So the D is on the y-axis here. So there are a lot of dots, and this is one of the caveats with D statistics when you have a large radiation like this. So it quickly becomes a lot of trios, which is so sort of complicating the interpretation here. And further complicating is the fact that these are not independent. So a single integration event can cause multiple positive D statistics. So basically what we've been trying to do is to systematically go through these, all these dots basically, and see if we could narrow it down and pinpoint introgression events in the evolution of guanons. So a logical way of starting would be to look at the strongest signals, the highest Ds that we found up here. And if we zoom into these dots, we find that the taxa involved in these D statistics are confined to 4 different species groups. So we have the Mytis, the Cephus, the Neglectus, and the Mona group, which we can then subset into a topology looking like this. And if we extract all the dots involving these taxa from the large plot that I just showed you in a more displayable way, we can see that in all possible scenarios of P1, P2, and P3, we get significantly increased D statistics. So D statistics is still on the y-axis here. So in principle, this would mean that we have signals of introgression in all possible combinations across these 4 different species groups. But we were able to, by looking at branch-specific alleles and excluding ancestral alleles, to show that the signal in B and D was only due to hitchhiking effects, basically. So in this approach, using this approach, we were able to say that the gene flow event responsible for these statistics was one single event occurring between the Cephus ancestor and the Mona ancestor, and it was bidirectional. And when we categorized this event, you can see that we've already categorized quite a few of the D statistic dots in the large plots, going back to this one that I showed before, by simply introducing one single ancestral gene flow event. So by systematically going through the D statistics in this way, we were able to identify 3 or possibly 4 major ancestral gene flow events. So the strongest one, the one that I just showed you between the Cephus ancestor and the Mona ancestor. We also found a gene flow event occurring at the very base of the guanon radiation, so between the bulk of the arboreal taxa and the bulk of the terrestrial taxa. And we found something also including the bulk of the arboreal clade, but being restricted to the Algocrocebus clade, and one event occurring up here between the Mytis ancestor and the Hamlini branch. And in addition to these ancestral gene flow events, we also identified 2 events among more terminal branches. So the one that I mentioned in the beginning between the Dryas monkey and the Pidgeotrychus clade. And we also identified 1 additional gene flow event occurring within the Algocrocebus clade here. So what is interesting here is if we recall the mitochondrial introgression, we find that there's actually quite little overlap here. So very few of the mitochondrial introgression events, or at least the transfers, seem to be having left traces in the nuclear genome. So this might be that you had a mitochondrial introgression occurring and then followed by a quick sweep without leaving any traces in the nuclear genome. It might be that we have incomplete lineage sorting playing a more important role here. So to sum this up, we find multiple ancient hybridization events throughout the evolutionary history of the guanons. And we also find that we have very weak connection between the nuclear and the mitochondrial introgression events. And with this, I would like to thank all the co-authors on this project and everybody who's in some way been involved in it. And thank you very much for listening.
First of all, I would like to thank the organizing committee for this invitation. It's a real pleasure to be here again this year. Today, I am presenting our work on the recolonization of the rainforest fragmentation in African trees with different dispersal abilities. This is part of a series of studies where we use genetic data of tropical trees, mostly lion trees, for understanding the biogeography of the rainforest. More recently, I have also started working in the Amazon. The rainforests are the oldest major vegetation type, but this doesn't mean that it's been an static biome at all. It's been the most dominant biome in between 56 and 33 million years ago, but it has been largely affected by the global trend of cooling and acidification of the climate. And this led to the extinction of many rainforest lineages about 33 million years ago. More recently, the rainforest has been severely affected by the significant oscillations of the climate that led to several cycles of contractions and expansions. To illustrate the impact of these oscillations on the African rainforest, this is the rainforest cover today. Is divided in two blocks, West African block and Central African block, separated by the Dahomey Gap. But if we go back 8,000 years to the humid period of the Holocene, we observe an expansion of the rainforest that connected the two blocks. If we go back 20,000 years to the last glacial maximum, we observe a contraction of the rainforest during these cold conditions that favor in favor of the expansion of the savannas. A main outcome of these expansions and contractions of the rainforest is the transition of tropical lineages in between the rainforest and the savanna. In this series of studies, we investigated these biome shifts in the legume tree Afzelia in Africa, and we detected two separate genetic clades for the rainforest and the savanna species that corresponded to two different fluid levels. This result indicates a single biome shift in the evolution of this genus in Africa. Another outcome of this climatic oscillation has been the fragmentation of the species distribution. In this study, we studied this genetic signal of rainforest fragmentation in 5 legume trees. The 5 species are widespread in African rainforest. 4 of them reach West Africa, and 3 of them reach the Congo Basin in inland. We use genotyping by sequencing. To avoid too many missing data, we sequenced 2 libraries per individual from 2 independent DNA instructions. And also, instead of using genotype calls, we were working with genotype likelihood for the downstream analysis, which were clustering, phylogenetic inference, and demography. Our results show for all species an early split of the West African populations from the Central African populations. This shows that the connection of the 2 forest blocks during the humid periods of the Holocene were not enough for an extensive genetic exchange between West and Central Africa. In addition, in Central Africa and also for all species, we observed a clear north-south genetic splits. And these splits do not correspond to any geographical barrier or any gap in the rainforest cover. We used data for dating these north-south splits and obtained divergence times from 3.4 million years ago up to 29,000 years ago. This result is highly congruent with the fragmentation of the rainforest during the ice ages, during the last few million years, in the Pliocene and Pleistocene. And interestingly, we observed much older divergence times in the short distance dispersed species and more recent divergence in the long distance dispersed species. This suggests that these species with highest dispersal capacity were able to erase this old signal of rainforest fragmentation. And also it shows that the dispersal capacity of each species is actually modulating how they respond to climate change. One of the main strengths of our study is that we do actually have accurate seed and pollen dispersal distances estimates. And we are only aware of 4 other studies of this type in tropical Africa. These estimates are not inferred from the fruit morphology, but they are properly modeled using genetic and spatial data of adult trees as long as seedlings and seeds. When the dispersal distance distribution is fat tail, it is indicative of long distance dispersal event, which might be rare, but evolutionary very significant, and is it illustrated in this study. Finally, for tracing the genetic signal of recolonization after forest fragmentation, we correlated genetic diversity with distance from refugia. And we use 3 different definitions for refugia. The classic last glacial maximum refugia of Malay and Anouf, which identify areas of refugia in areas of high species richness for Malay and high precipitation for Anouf. And also we estimated a last glacial maximum refugia for each species using climatic modeling. We obtain significant diversity declines with distance from refugia for the long distance dispersed species only. For Pericopsis, we observe higher genetic diversity closer to inland refugia, and for Disseminantes closer to coastal refugia. This suggests that the 2 species survived in different refugia. And also, it's very likely that we are observing here for this long distance dispersed species is a genetic signal of recent expansion. Finally, I would like to thank my co-authors. We are an international team based at the University of Copenhagen, the Free University of Brussels, the University of Exeter and Kew Gardens. And also I'd like to thank all the scientists, all the laboratory staff and all the fieldwork staff who have greatly contributed to my project. Thank you.
Good day, everyone. My name is Blessing Machuku. I'll be presenting on the topic structural and respiration profiles of microbial community from sunflower plant soils. Sunflower is from the family Compositae, an important oilseed crop predominantly used worldwide for the production of cooking oil. In South Africa, sunflower is currently the 3rd largest grain crop cultivated after maize and wheat. Amplicon sequencing is a non-culture-based approach to collectively study the composition and structure of entire microbial community in an environmental sample. The community-level physiological profiling offers an easily applied protocol yielding information regarding mixed microbial community and functional adaptations over time. Plants grow promoting rhizosphere bacteria. PGP are a group of bacteria capable of actively colonizing the root system or the root region and improve plant growth and yield. Methodology. Soil samples were collected from 2 sunflower farm sites, which are Shila and Isoseng, which were located at Shila and Isoseng in Northwest South Africa. Soil samples were collected and taken to the lab and stored. The soil samples were also sieved to remove pebbles. DNA extraction was carried out using DNA extraction kits, Zymo to be specific. The purity and concentration level were determined using the nanodrop spectrophotometer. The extracted DNA was sent to Molecular Research Laboratory, Mr. DNA in USA for Illumina sequencing. Functional measurements using community level profiling and physiological profiling, CLPP technique, were used to assess the ecosystem function. A total of 11 carbon substrates, including methionine, tyrosine, galactose, maltose, fructose, glucose, and sucrose, were all used for the functional analysis. The quantity of CO2 respiration per gram of soil per well were calculated using the formula in the microRESP manual. The individual substrate respiration were subjected to statistical analysis, statistical processing, and statistical analysis. Preliminary data that was collected from Mr. DNA was uploaded in MGRAS. Risk quality was done by performing quality check of Illumina sequence using FASTQ. Final operational taxonomical units were taxonomically described using BLAST and against a corrected data result from RDPII and uploaded on MCBI, which has the bio project number as presented on the slide. The species richness was determined using the rate refraction, and the alpha diversity were depleted with the aid of Shannon, Chow I, and evenness indices. Krasowale's test was used to describe the diversity indices across sites. Shining heat map was used to plot the relative abundance of microbial species at phyla level. The relationship between the physiological parameters were ascertained by one-way ANOVA with the Turkey HSD test with the aid of employing SPSS package, p-value less than 0.05. Table 1 shows the soil properties from sunflower rhizosphere and bog soils. The means in the same row with the similar alphabets differ significantly, p-value less than 0.05. Figure 1. Figure 1 is the sequence tags where the sequence tags were assigned to Sheila and Cripan rhizosphere and bog soil samples in different taxa through metagenomic rapid annotations using subsystem technology. The relative abundance of the microbial phyla for each soil samples as presented. The row Z-score shows the color intensity and the abundance of microbial species on each soil samples. From the figure, you can see that gamma protobacteria was more abundant in Sheila bog soil, while bacilli was more abundant in Sheila rhizosphere. In Sheila rhizosphere, we can see that acidobacteria, flavobacteria, alpha protobacteria and beta protobacteria were more abundant in Sheila rhizosphere soil, while the cytophagia was more abundant in Cripan rhizosphere soil. Figure 3 is the QLPP of microbial community measurement using the microplate. By totaling the respiration responses of the entire 11 carbon substrates used for the study, we were able to measure the total soil microbial activity. The rhizosphere, when related to the bog soil, had total substrate responses that were not significantly different. Influences of soil type on soil functional properties as measured for distilled water which serves as the control. The figure shows that the respiration rate of individual carbon substrates from the slides, SE, which is the standard error, NS, not significantly different, S significantly different, while double X shows more significantly different. Our results show that, that generally, some of the rhizosphere soil always have significant difference higher in soil respiration and capacity to degrade carbon substrate than in bulk soils. Significantly, methionine had a more significant difference between rhizosphere soils than in bulk soils. The figure 4 also shows the influence of carbon substrate on the relative abundance of microbial species as microbial species. And from this slide, you can see that there were no significant difference between the soil samples. Also, from figure 5, there were no significant difference for in the soil functional properties of maltose, fructose, glucose, and galactose. Conclusion: this research reviewed the respiration profiles of microbial species from sunflower soils. With the use of the microREFS techniques, we measured the community level profile of microbial functionality in sunflower soils. We showed that soils from sunflower had the capacity to degrade carbon sources due to the higher soil respiration when related to their bulk soils. Our findings have important implications, especially in soil ecosystem, for understanding how sunflower microbial community activities shape the soil microbial functionality and the potential soil ecosystem functions. These achievements will positively contribute to offering solutions to food security problems prevalent in developing nations of the world. I would like to appreciate National Research Foundation, South Africa, and the World Academy of Science for a stipend, which is of great help for my PhD study. Dr. Ayoshina would also like to thank the Northwest University for the postdoctoral fellowship. My supervisor, Professor O.O. Babalola, would also like to thank the National Research Foundation for a grant that supported research in our laboratory. Thank you for listening.
Thank you very much. So a lot of the research in my lab centers around the question of how complex landscapes drive biodiversity within species. So for example, widespread species will experience a great deal of variation in their environment, as well as evolutionary history of local population that can leave unique changes in DNA sequences, as well as variation in things like gene expression that can ultimately influence how organisms respond to environmental variation, and leave variation in things like functional traits and phenotypes. A lot of the work that we do centers around the Sierra Cascades as a laboratory for studying complex landscapes because of the great deal of environmental variation associated with latitude and altitude in the system. So for example, we can sample populations across latitudes and look at large-scale gradients and say latitude temperature associations. We can also zoom in within latitudes and sample populations across elevations and thus get at regional or local scale environmental changes nested within this broader biogeographic latitude gradient. The work I'll be talking about today centers around two common species of bumblebee, Bombus vazenskyii and Bombus vancouverensis, which are common throughout the region. As you can see from the range maps here, they occur across both a wide range of latitudes and elevations within the system, and we have populations that we've sampled across latitude and elevation gradients across this region. So one of the questions that we often have in our work is there signature in the genome of these complex landscapes driving environmentally induced natural selection or local adaptation? And one of the tools we use to do that is genome environment association analysis. The basic idea here is you sample individuals across a species range from a range of different environments at different spatial scales. You perform sequencing to call genetic variants, in this case, single nucleotide polymorphisms. In the talk today, I'll be talking a little bit about whole genomes, which we have for over 100 individuals of each species sampled across latitudes and altitudes. I also have a little bit on genome reduction sequencing using rad tags from a finer scale of hundreds of individuals. Once you have genetic variants, you identify how SNPs are associated statistically with the local environmental conditions using methods that control for neutral population structure driven by genetic drift and gene flow. Just to give you an idea of what this looks like, here's an example of a genome environment association analysis conducting using 122 whole genome sequences from Bombus vancouverensis. And what we see here are significant signatures of unusual allele frequency shifts associated with elevation after controlling for population structure for scaffolds across the Bombus vancouverensis genome, with these red peaks indicating those unusual associations. So what you can see is these red peaks are sort of scattered throughout the genome, and they fall in interesting regions in many cases, such as genes that are associated with motor neuron development, as well as genes that are associated with hypoxia. We also performed similar analysis in Voznesenskii. Unfortunately, we don't have the whole genome data set analyzed yet, but we do have over 600 individuals sequenced using that RAD-seq method. And one of the things that we see is that this species actually doesn't have as many strong signatures of environmental association as we see in vancouverensis. However, when we look across the genome, we do see a number of regions of the genome that are associated environmentally with various thermal variables. So there is some signature of local adaptation across this species as well, including in interesting genes that are associated again with motor neuron development, general neural development, and ion transport. And several genes in this category in particular have been shown to be associated with cold tolerance in other insects. So we have sequence variation that shows these unusual statistical associations with spatial environmental variation across this complex landscape. But the question remains, do these complex landscapes and environmental variation drive actual changes in functional traits that influences how organisms thrive and survive across these complex landscapes? So to answer that kind of question, we'll focus again on Bombus voznesenskii here, which is a nice species because we can sample it across a range of thermal environments, from cold to hot environments, and we can do so in a way that decouples that environmental variation from space by sampling across low and high elevations at both low and high latitudes. So what we did is we sampled queens from these populations and brought them back into the lab and established common garden laboratory colonies. Using methods developed by my collaborator Michael Dillon's lab, we measured thermal limits in these species. So we measured critical thermal limits at both upper and lower temperature extremes to measure how populations varied across these across the species range. What we see is that upper limits do not vary among populations. All the workers do the same thing regardless of the region of origin. In contrast, lower thermal limits vary strongly by climate of origin, such that common garden workers derived from queens sampled in cold environments are able to withstand much colder temperatures than workers derived from queens sampled in warm environments, thus suggesting in these common garden populations that there is some evidence potentially of variation, local adaptation in cold tolerance. We also performed RNA sequencing of all of these experimental bees. Just for the sake of time and simplicity, I'm going to show you a single top co-expression module that's sort of a group of genes that's doing all the same thing. It's the most significant one in the data set that has 111 genes, resulting from one of these WGCNA network analysis. So when you look at bees that were ramped up to their thermal extremes at the upper limits, similar, paralleling those patterns that we saw in the physiology, there's no variation among populations. There's a large response in gene expression across populations, but it doesn't vary by region. In contrast, when we look in the control, and especially in the CT-min bees, we see this variation regionally, where you have the populations from the cold environments, their genes are doing something quite a bit different from the populations in the hot environments, suggesting that there's both regional variation across the species range in both physiology and tolerance to cold, as well as gene expression at both baseline and under cold stress. So what drives this variation among environments? One of the things we're starting to look at now is a possible role for epigenetic processes. This is still very preliminary data. We're just starting to analyze these data now, but we sampled, again, workers of Voznesenskii collected from the cold extremes and the hot extremes of the species range. We looked at one kind of epigenetic process, whole genome methylation profiling using bisulfite sequencing. We removed all the single nucleotide polymorphisms detected in the whole genome dataset. And what we see is there is in fact regional clustering of global CpG methylation, especially in the high elevation populations, which if you remember were those populations that had the broadest range of physiological thermal tolerance as well as gene expression tolerance under environmental extremes. Now, we don't exactly know what methylation does in insects with respect to gene expression and phenotype. So this is still very early days. We don't know if methylation is affected by thermal tolerance, thermal exposure, drives thermal tolerance, or is this solely an effect of ancestry? So we're currently working on these epigenetic processes moving forward. Finally, what the ultimate goal of all of these things is to integrate all this genome data to make predictions about climate change sensitivity in this organism, right? So just to give you an example, we can take regions of the genome that are associated with spatial environmental variation. We can project those associations into climate space represented geographically, and then we can project that into the future under climate change and look for those spatial locations where there's going to be a breakdown in the environment genotype association, and look for those regions of the gene space that have unusual frequency, genotypic information that's likely to be lost under climate change, and thus maybe can be conserved to maximize adaptive potential. And with that, I'd like to acknowledge everyone in my lab and my collaborators and my funding sources. Thank you very much.
Hi, I'm Kate, and today I'll be discussing our investigation of neutral and functional diversity across the entire range of the Tasmanian devil. So what do we mean when we examine genetic diversity? Well, firstly, neutral markers are typically used as a proxy for genome-wide processes, such as inbreeding. And these days, SNP datasets can be generated with relative ease at a large scale across multiple samples. And although some SNPs may be functional, typically, these methods do not target functional regions of the genome. And that has come under recent criticism in the literature, as neutral diversity does not predict adaptive potential. And in fact, it is the loss of functional diversity that matters, particularly for species facing selective pressures. But if we are to examine functional diversity, how do we select which traits matter? Well, current methods tend to use association-based analyses to identify putatively linked SNPs from neutral datasets or genome-wide datasets. Or researchers can choose to focus on a particular gene or a few genes of interest, such as the MHC in disease studies. But by managing for a particular gene of interest, we risk losing diversity in other genes that may be equally, if not more important, for the adaptive potential of the species. So the Tasmanian devil is an excellent example to investigate neutral and functional diversity, as it has undergone large selective pressure since the emergence of devil facial tumour disease, or DFTD, in the mid-90s in the northeast of Tasmania. And this has since spread across the state west to almost the entire state. And to give context, Tasmania is about the size of Florida or Ireland. So as a result of the transmissible cancer, an insurance population was established in 2006 to protect the species from extinction. But recent evidence has shown that some devils in the wild may be evolving resistance to the cancer. And so it's unknown whether releasing devils back from captivity to the wild could impact evolutionary processes. In terms of the genomic resources available, a reference genome was published in 2012, and this has since been updated with long-read technology to a chromosome length assembly. So the aim of this study was to comprehensively assess the patterns of genetic diversity across the species range, to examine how well the insurance population reflects the genetic diversity of the wild, a key consideration for captive populations, to assess whether functional diversity reflects genome-wide diversity, which is currently used to manage the species, and therefore inform future management of the devil. Our geographic sampling was comprehensive, as can be seen in this map, where the point size is proportional to the number of samples. Overall, we sampled more than 1,000 devils, and these samples were collected from field monitoring trips, the captive insurance population, a citizen science project where members of the public reported devils that had been killed by cars, and these were opportunistically sampled where possible, and a crowd-funded field trip to the southwest, which is a remote area of Tasmania that was previously had not been surveyed for Tasmanian devils. We use two genotyping methods. Firstly, DART-seq, which is a reduced representation sequencing method where the genomic DNA is digested with restriction enzymes, and these fragments are sequenced to generate a SNP data set. Other studies have shown that these SNPs rarely fall in functional regions, and so for the purposes of this study, we considered this to be a neutral data set, and we obtained over 1,000 SNPs. To examine functional diversity, we designed a custom target capture method making use of the available annotated reference genome. By doing so, we were able to examine functional diversity at over 500 genes, a much larger scale. And these genes included ones involved in immune processes, reproduction, and loci that had previously been identified as associated with DFTD. And so hybrids baits are designed to hybridize to the target genes of interest. A magnetic bead is then bound, and the magnet is used to separate the target DNA from the non-target DNA, which is washed away. And the target DNA is then amplified and sequenced. We obtained 195 SNPs at 123 samples. We used PCOAs to examine diversity across our data set. In the DART genome-wide analyses, or in all PCOAs, each sample is represented by a dot, and the closer the samples are together, the less variation between them. As we can see, the genome-wide data reflects fine-scale population structure, very similar to that of the biogeography of Tasmania. The functional data, this is not so obvious. However, there are still patterns between the northwest, northeast, and southwest of Tasmania. Comparing the insurance population, shown here in orange, to the wild, shown here all in grey, we can see that the captive population does in fact appear to reflect the wild diversity, perhaps with the exception of the southwest, which has never been brought into captivity. And importantly, it still appears to reflect the functional diversity, even though they'd be expected to have different selective pressures in captivity. At the DFTD-associated loci, of which there were 51, there was no real geographic structuring in the data set. However, the insurance population, importantly, still appeared to reflect the wild, even though they're not directly exposed to DFTD. Examining the genes driving the variation between the eastern and western populations, of particular interest were genes involved in male reproduction, of which sperm function contributed the most to variation. This is of interest as we have previously observed a reproductive decline in a population with no DFTD in the northwest of Tasmania. So it's an area of further investigation. Finally, we examined diversity statistics such as standardized heterozygosity on the left and the inbreeding coefficient, FIS, on the right. Observed heterozygosity was lower in the Western populations than the Central and Eastern populations, although this may be an artifact of the individual used to generate the reference genome. An inbreeding analysis identified multiple populations that may be a target for future actions, such as translocations and genetic rescue, some of which is already underway. So in conclusion, our analysis revealed fine-scale structure across the entire species range of the Tasmanian devil. We demonstrated the utility of a reference genome for examining functional diversity at a much larger scale than had previously been done. And by doing so, show that the functional diversity generally reflected the genome-wide patterns. And importantly, the insurance population reflected the wild diversity, including at the DFTD loci. Further investigations of genes of interest is ongoing and haplotype frequency analysis, and this will inform future management such as wild-to-wild translocations. And finally, I'd like to thank the members of the Australasian Wildlife Genomics Group, as well as our funding bodies and collaborators for their support of this project. Thank you.
Okay, thank you everybody for those great presentations. Nina, do you want to start, or shall I start with asking a question? I guess Matthew, well, Webster asked Jeff, said that's very interesting work. Do you think any of the genomic regions that associate with elevation are inversions?
So that was actually one of the first things we looked at. And so what we do see is that there are structural variation in some of these regions, but it doesn't seem to be one of these situations where there's inversions flanking these regions of differentiation. And I think what we're actually seeing is very similar to what you guys saw when you analyzed the high elevation bumblebees out in Colorado population with the Bombus sylvicola, where we're seeing that there may actually be associations with some of these structural regions of the chromosomes near telomeres and centromeres that may be harboring some of these regions due to low recombination. So what that means is
interesting. Thank you. Then next we have a question from Pam Saltes to Rosalia. She's asking, where are the genetic patterns that you have identified for species of trees in Central Africa showing the effects of ancient geographic splits also observed in other species that occupy the same regions? It would be interesting to know if these are region-wide patterns.
Okay, yeah, thank you for the question. It's actually very interesting because, yes, this seems to be a recurrent biogeographic pattern. So this split that we are finding between the northern and southern populations in Central Africa and the Central African rainforest, despite the fact that the rainforest cover is continuous today, it's also found in many other tropical trees that we have investigated in the research groups I have been working with, and some other groups. But also it's very exciting because these north-south splits are also showing up in some studies on frogs and also on reptiles. So it seems to be a recurrent pattern that is just coming up right now that shows the effect of the fragmentation of the African rainforest during the ice ages.
Okay, thank you. So I'll ask a question to Axel from Nina. So are the introgression events that you see, that you've detected between lineages with highly divergent karyotypes, or, picking up also a comment from my question, what's the level of divergence that you can, between the genomes when the introgression took place? Do you have a sense of that?
Yeah, so thank you. I mean, the introgression events that we've detected ranges actually across different sort of differences in karyotypes. The very recent introgression events that we've detected, obviously they occur across more similar karyotypes, but the strongest sort of ancestral gene flow event that we detected between the Cephus and the Mona group. So there, the Cephus group is generally have diploid chromosome numbers around 66, while the Mona group is slightly over 70, usually also variation within the groups. And the further back you go, obviously it becomes more and more difficult to establish what the karyotypic differences were at the time. But there are also presently documented hybridization events occurring across karyotypes. So it seems that this is not a fixed hindrance, at least. And in terms of nucleotide divergence, I haven't actually considered specifically the nucleotide divergence at the timing of the hybridization events. We have been exploring a little bit about using non-admixed sister lineage to sort of try and polarize the exact timing of the introgression events. But I suppose we could also use that timing then to look specifically at the nucleotide divergence. It's a very good question.
Great, thank you. We're almost out of time here, but I'd like to also ask a question for Kate. So I was curious, in that set of targeted functional variation that you've analyzed, do you think that fully captures what might be under selection in response to the adaptation to this to coping with this cancer, or do you think it's possible that there could be some narrow signals of selection in other parts of the genome that would be important to monitor as well?
Yeah, so there'd certainly be other parts of the genome as well that could be involved. So our target capture did mostly focus on the immune genes and the reproductive genes, which are also associated with, we have seen evolution towards devils reproducing younger in DFTD associated areas. So it's quite possible that some of the genes we looked at could be involved or are involved with DFTD, but there would likely be many others as well. And of course, it's the ones that we selected are based on previous research, a lot of previous research that has gone into annotating the devil genome and identifying these regions, and also the cancer research that has looked into genes of importance. So I guess the target capture is a good balance for wanting to look at functional regions without necessarily having whole genome data for such a large sample size.
Okay, well, thank you. I think we're pretty much out of time. I wanted to thank all the speakers again, and also remind you that there are posters to go and look at, and you can partake in the spatial chat and meet up with poster presenters and speakers and others. And also that there's a second session in this this theme tomorrow evening at Stream C at whatever time zone you are, at 6 o'clock UK time. So I hope to see you all there.